cytometry core facility. You'll actually see the core facility right here in this slide. We are located down in Bloomington, Indiana, and we are in the biology building and located in the basement. I think where several service cores tend to be located. But we do serve all of the CTSI schools and locations and offer various services related to flow cytometry. And I do want to thank also the Indiana CTSI Access Technology Program for this opportunity to host one of these sessions. And I'm just going to also just show you, if I can get to the next, advance my slide. There we go. And just to let you know, so your presenter today is going to talk about the SH800, and I'll introduce him in just a moment. I do want to mention that our SH800 was supported in part with support from the Indiana CTSI and funded by a grant number ULTR002529 from the NIH. So we also are grateful for this opportunity because we did receive funding for the SH800. So this is the SH800 instrument. Our instrument is a 2-laser, 8-parameter, 6-color instrument, and users can be trained on the instrument. And I will also mention that this is our contact information. So, contact information can be found on the facility website. So if you go to the facility website, you'll find my email and other information about the flow facility. So now it is my pleasure to introduce Jerry Altz with Sony Biotechnology. He is the Midwest Sales Specialist, and he is going to tell you more about sorting with the SH800 for single-cell transcriptomics. Thanks, Jerry.
Thanks, Christiane. Can everybody hear me all right? Okay, here we go. So today we'll talk about using Sony Cell Sorters for sample prep in single-cell transcriptomics workflows. As Christiane said, I'm Jerry Altz. I'm the Midwest Account Manager for Sony Biotechnology, and I've been with the company for about 1.5 years. So, what makes the SH800 uniquely suited to these sorts of workflows? Why does sample quality matter? How can cell sorting be used for sample optimization? And what applications have Sony sorters been incorporated in terms of transcriptomics workflows? So these are the questions that we're going to address today. Let's start with the instrument itself. Christiane gave a quick glance at it, but we'll dive in a little deeper. Going through an overview, the system basics. We'll kind of skip over biosafety and then deal with the optics, the fluidics, the options, and the software. So the benchtop SH800 cell sorter permits sorting of a wide range of cell sizes and applications. We have 3 what we call microfluidic sorting chips. These chips function as both the flow cell and the nozzle of the sorter. We have 70, 100, and 130 micron sizes. They're fully integrated with comprehensive fluidics controls, and the advanced automation setup, acquisition, sorting, and analysis makes this easy to use, as well as the intuitive system software that supports sorting into a wide variety of receptacles from Eppendorf to Falcon tubes, and actually even 6 to 384 well plates, though most commonly people use 96 well plates. So it's a plug-and-play design meant for a benchtop fit. It can be incorporated into any lab. We see here the instrument itself and the computer that runs it. There's also a fluidics cart that we'll see in some other slides, and an air compressor. So basically what makes this system very easy to use is that it has multiple sensors and cameras all throughout, measuring every mission critical component of the system. So here we see a basic layout of the laser setup and the filters. So you can equip the SH800 with up to 4 lasers, from violet to red. It's field upgradable, so Christian, if you feel like adding any more lasers, Sony is happy to help. And the automated daily alignment of that sorting chip to the laser is really what ensures consistent results from day to day. Basically, you start every day just as if a technician from the company had come in and given your instrument a tune-up, as opposed to fixed nozzle systems where, you know, the day after the yearly overhaul, you slowly start sliding out of spec. So in terms of the detectors, it's freeform PMT layout. They're user exchangeable, and we'll see a nice shot in a couple of slides about the optical filters and how to access them. But it's quick and easy to custom configure the instrument for whatever sort of experiment you're running, from looking at fluorescent protein to immunophenotyping. As Christian said, it has up to 8 parameters, so forward, side scatter, and then 6 fluorescent channels. Here's a close-up of the microfluidics sorting chip. We see at the top a port for the sheath fluid, in the center a vacuum line, and then the lowest port toward that V at the bottom there is where the sample actually comes in. You can see that channel is actually a little bit narrower than the rest of the chip. This is so if a clog does happen, we want it to happen there right below the sample port, so that a simple backflush can clear it. Meanwhile, the sheath fluid coming in from that top port continues to flow out the nozzle unimpeded, and so your drop drive and break off, none of that is affected. A clog does not mean that you'd have to recalibrate or anything. And so like I said, we have 3 different sizes: 70, 100, and 130 micron. So there are very few cell types that wouldn't be a good fit for those sizes. I know people do sometimes ask us about intermediate or larger sizes, but those are the 3 that Sony has for now. And so we just see the fluidics system. The sheath fluid is well filtered, and that's all monitored. The sample flows to the chip and out the bottom. So the sorting chips were, like I said, designed for use with a wide range of cell sizes. Their daily alignment in the calibration process really makes this instrument so much easier to use, as opposed to earlier systems with fixed nozzles where there was a lot more manual input required to calibrate the instrument. With Sony, it's basically a push button process. So, and as I said, you can easily recover from clogs and reuse the chip using software options. So, and it does quite a lot toward stopping any sample carryover. So once you scan the QR code on the little envelope that the chip comes in, you have 24 hours in which you can use it for as many samples as you can fit in. After that, it has to be discarded. We do this for 2 reasons. First of all, continuous use of the same chip. Even with cleaning, you will eventually get accumulation of salts and protein in the channels. Also, given a long enough time frame, talking like months to a year, the energy of the 405 nanometer violet laser will actually start to etch the plastic at the interrogation point on the chip. That's not a problem in daily operation. And whenever you get rid of the chip, at the end of the day. But yeah, if we kept trying to use the same chip over and over, that would eventually be a problem. And there's the laser interrogation site there at the bottom, right before it comes out the nozzle. So as I said, each chip is serialized with a QR code. It can only be used for 24 hours. The chips cost about $32.75. We could have made them with optical glass so that you could use them for much longer, but they would cost a lot more. So they were made to be disposable. So in terms of the specs on the chip, they run at about half the pressure of an ARIA. So at 70 micron, you're running at about 40 psi. We're not as fast as some of the fixed nozzle systems. You top out at about 12K events per second. For the 100 micron, the pressure halves, so it's down to about 20 psi, and you can run about 7,500 events per second. And then the 130 micron, we typically would reserve that for like stem cell or neuronal work, basically applications where you have to be just a little more gentle. And there you're running at 9 psi, and about 3,000 events per second is the max that we would recommend. So I had promised a good shot of the optical filters. In this photograph, underneath the woman's right arm there, you see a number of colored tabs. Those are the optical filters. You can pull them right out with your fingers. You don't need to go searching for a little Allen wrench or screwdriver. And the system will prompt you to see if the filters are in the place that you would want them to be for your particular experiment. That happens when you do the startup. So, but the new sorting chip provides a clean nozzle each day, really enables the automation of the setup. It's easy to exchange, so you just feed it in like a disk into a Blu-ray player. And it's low carryover and very easy to declog. Even the sample line, if you're doing that last experiment before defending your thesis or before publication, and you absolutely want to ensure that you have a totally pristine sample path, you can change the sample line again just with your fingers. Sony never wants you to have to keep around, like I said, a little set of Allen wrenches or special tools. But and then do the calibration, and away you go. So in terms of the options for sorting devices, it's a two-way. You can switch over very quickly, though, from sorting bulk sorting into tubes to sorting into plates. So you see the white plastic holder just rests on that metal arm, and there's one big finger screw at the bottom. And so you can take your tube holder out and then screw in this arm. You can also see at the left of the arm a little metal knob in the floor of the stage. That's the Peltier coupling. So it can heat or cool your samples. Both the deposition and the sample chamber can be heated or cooled to improve sample viability. So we can sort into any number of tubes from 1/2 mil Eppendorfs on up to 15 milliliter conicals. I always call people's attention to one little thing there. The Eppendorf holders, that funny little tab at the top of the holder, that's just there to hold the cap out of the way. 20 years ago, in my own time at the bench, we had to snip the caps off with scissors to make sure that we didn't ever bend our sample probe. But the reason I point that out is just, if Sony gave that much thought to the design of the tube holder, you can rest assured that the more critical components of the system, from the fluidics to the optical bench, are definitely thoughtfully designed as well. So all the tube holders can be cooled directly with the Peltier coupling. If you're using the plate holder, first of all, we have holders for just about any kind of plate you want. They have gel packs inside them, and so you would have to put the holder into an incubator or into a refrigerator to heat or cool a plate, because when it's perched up on the sample or the plate holding arm there in the deposition chamber, it's not making contact with the Peltier coupling. So the software is very user friendly, especially the automated system calibration. So it goes through 5 steps, but it's precise and accurate, and it's streamlined for all users. So you don't have to be a flow jock with a ton of experience to to get this instrument well set up for an experiment. Even a first year, even a first day student can go ahead and follow the prompts and hit the buttons. Here's a screenshot of what it looks like when you're doing the system calibration. You can see the calibration beads. It's a cocktail of 3 micron and 10 micron beads. You can see the droplet break off. And as the system progresses through each step, that little green bar at the bottom will fill in. And then when you're done, it'll say calibration successful, ready to go. So the key benefits, though, of the SH800 are its ease of use, I would say, is the biggest one. Because so much is automated, not just the calibration, but even compensation. There are wizards. There are also wizards in the software to help you with your sort controls as well. One nice thing about the SH800, though, and Sony instruments in general, while we're able to deliver that sort of push button ease of use, it does not come at the cost of more advanced features. So if you want to do your own compensation, or you feel like you're being forced to ride a bicycle with training wheels on it, no, that's in our software. There's always the ability to fine tune, like I said, everything from your compensation to even your droplet controls or your sort controls. We can discriminate down to a quarter of a droplet. So this is a very capable instrument. Don't be afraid of it at all. It's very easy to use. And yeah, it's very flexible as well. So quick glance at the software. You'll notice that it looks a lot like other software that you've used for flow cytometry in analysis or in sorting. It also bears a strong resemblance to Microsoft Office's productivity suite. That was done very deliberately, just to sort of leverage that mental sort of muscle memory. At this point, we've all used Excel and PowerPoint for so long that we're used to navigating the ribbon of tools at the top and the different tabs. So first you create an experiment, create plots and stats, perform compensation, acquire data, and sort. And so on the first page, setting up your experiment, again, it looks very much like other software that you've used. You can plug in your experiment name, date, select the channels that you're going to use. So here's the main page of the software. From here, you can run compensation controls, preview gate target populations, which is sort of a neat function. So you can see what a new gate would, gate would look like before you create it, and then set up the collection device, and the sort controls live at the bottom. And if you look closely at the top left, you can see the little blue box that's file, and you can go back and save, and then experiment, cytometer, compensation, and worksheet tools. And so as you click through from one to the other to the next of those tabs, the tools in that top ribbon will change, just like they do in Excel or PowerPoint, or like I said, other Microsoft Office productivity software. In terms of the compensation, spillover in detectors can be subtracted and corrected by creating a compensation matrix using single stain tubes. But like I said, you can also do it manually. We don't make you ride the bicycle with training wheels. You may not like the suggested compensation that the software provides. You're free to create your own. So, but here's what it looks like when you use the wizard in the software to do your compensation. All of the wizards pop up like a black screen like that at left. And then basically it just tells you to feed through the tubes one at a time, and to make your adjustments until your peak is in the first decade of your plot, and away you go. And just a quick look at some of the data that was acquired with the SH800. Uncompensated here first. I know it looks kind of ugly, especially to those of you who are doing flow on a regular basis. With the compensation added, though, we get rid of those smears and rockets, and we start to see nice, much cleaner populations. So the wizard does a pretty good job with the compensation, but like I said, you don't have to accept what it tells you. You're free to do your own thing. So in terms of acquiring data, the controls, like a lot of other flow software, are on the left. You can see the start, stop, record buttons there. And then you can sort into plates or tubes. And in the sort control panel at the very bottom there, you can see sorting method. And here we're two-way sorting into tubes, but that's the menu you'd use to change into plates or any other container that you want to sort in. And then there's a function in the software as well to aim your droplets. It's especially important if you ever did have to use a 384 well plate. The 96 well plate is pretty forgiving. It's a larger target, but you can leave the lid on your plate and then fly down about a dozen or maybe 20 or so droplets, and that'll make a wet mark on the lid of the plate big enough to see and make sure that you're hitting the bullseye rather than off toward the edge of the well where your cell might splat out against the side of the well before it has a chance to land into whatever buffer you're sorting into. So in terms of plate sorting, you'll notice you recall from the previous slides, the plate holder looked a little bit canted. So that's actually on purpose. The stream does not emerge perpendicular. It's actually at a slight angle, and so we angle the plate so that the droplet path it's encountering the plate at as close to a 90-degree angle as possible. So a neat function that we have in the software is index analysis. So it records the XY coordinate of each sorted event. And basically, it allows you to track the phenotype of individually sorted cells. You can annotate cell data, and that is important for single cell genomics and cell cloning, as we'll get to here as we finish up with the instrument. But basically, either way, you can click on wells in the plate, and it'll show the actual event within your dot plot within your gate, or you can click on a dot in the plot, and it'll show you what, if any, cells in your plate match that phenotype. So yeah, here we go, the well select mode. And like I said, you can see either by the wells in the plate or the dots in the plot exactly where your different cells landed. So, and then you can also draw a larger gate and see what cells you may have collected that meet that phenotype. So, in terms of data export, we're pretty flexible. We can export comma/value files or FCS 3.0 or 3.1. And one nice thing about Sony is that anybody who wants to can have a copy of our software for analysis. We never want somebody to be stymied, standing there with an ice bucket under their arm waiting to run a sample while a colleague is on the computer connected to the instrument analyzing data that was acquired the day or the week before. So everybody can take the software and put it on their own laptop or whatever device and use it at their leisure. So, and then here's just another shot of the indexing. This is a feature that I'm not aware of other manufacturers having, so we're pretty proud of that. But in summary, the automation makes this so easy to use, from the calibration to setting up your sorting and monitoring the system along the way. The guided workflows ensure that Anybody from a first-year student to a very experienced user can use the instrument and turn out the same results. With the sort modes, standard and custom modes, basically there's not a whole lot of applications you can bring to this instrument that isn't ready to handle. So the index analysis, annotating single cell sort data for correlating phenotype with functional genomic assays, makes this well suited to incorporating into your single cell genomics workflow. And then in terms of the maintenance, it's not something that we touched on a lot, but it's very easy to clean up the machine after you use it, and to to put it away for the night and make sure that it's ready to go for the next user. So getting to the heart of the matter, the single cell genomics that the SH800 has been used for, one of the first questions to address is, why does sample quality matter in single cell transcriptomics? So whether your cell source is tumor tissue, organs, blood, cell line, you're harvesting the cells and mechanically or chemically dissociating them, two issues can arise. You can have free unbound DNA and RNA and unreliable cell recovery. And then you can have a heterogeneous population, depending on the sample type you started with. And if you have few target cells from the input sample, it can reduce your throughput. So usually single cells or nuclei are isolated from a bulk sample. You do barcoding and library construction, RNA sequencing, analyze and visualize the data. But less than 10% of the sequencing reads are associated with intact cells or nuclei if you haven't taken any steps to clean up your sample. And so basically you can wind up with really rotten signal to noise ratio. However, if after you get your homogenized tissue, you purify viable cells or intact nuclei by cell sorting with the SH800, you can continue with the rest of your normal workflow. And then more than 75% of the sequencing reads are associated with intact cells and nuclei. So basically, you get a better signal to noise ratio, and you're able to increase the specificity of your analysis, because you're not looking at cells that you don't want. The cell sorting has been used for sample optimization in single cell transcriptomic workflows across a variety of applications, from cancer, genomics, metabolic disorders, infectious disease, looking at the immune repertoire. That's something that we'll get to in a few slides, system biology, and creating cell atlas. So it's like I said, a very flexible platform that has been used in just a large variety of applications associated with transcriptomics. So here again, we see just a picture of the inside, the plate holder or the tubes. So whether you're going on to do drop-seq or smart-seq assays, we're set up to help you optimize your sample before continuing on your workflow. So for ATAC-seq nuclei isolated mechanically or enzymatically, you can wind up with a prep that's like 85% debris and 15% nuclei, which is far from ideal. But if you stain and sort just with viability dye, you can identify intact nuclei and get to a much cleaner sample. So here we show about 85% of the sample is actually the nuclei you want rather than the debris you don't. So the Sony SH800, as well as its bigger brother, the four-way sorting MA900, have been used for single cell transcriptome profiling of patient-derived xenografts. And so basically the intratumor heterogeneity poses challenges to RNA profiling. And you've got infiltrating lymphocytes and other things in there. So when the sample isn't enriched, the sequencing reads are pretty noisy. And so we're able to improve the sequencing resolution by sorting for, in this case, I believe CD8 T cells. Yeah, here we go. So viable tumor cells were sorted to remove the infiltrating lymphocytes, and the sorted patient-derived xenograft cells were loaded on 10x Chromium platforms to profile the transcriptomes associated with clinical outcome. And I said we've also done multi-omic assays for immune repertoire analysis. So in this case, a sorter was integrated into the workflow. The complex sample was PBMCs. They were stained with total seq antibodies, antibodies to surface markers, and then a dextramer panel for VDJ profiling, as well as a viability dye. And so they were run on the sorter and then gated for CD8. And so basically by cleaning up this sample, we can get the gene expression of all the different cell populations present, rather than a simple average. And in this case, the purified target population, I think we wound up getting to 98%. 98% were multiple bound T cells that could be used for IDing the T cell repertoire, as well as antigen-specific T cells. So in summary, just the automation of the system, this cell sorting system, it can be seamlessly integrated into your single cell transcriptomic workflows. Cleaning up your sample ahead of time can give you just so much better results. So, but with the fluidics path being disposable for a matter of about $250 and less than an hour of your time, you can remove every portion of the sample path, replace it, recalibrate, and be ready to run. So there's no sample or DNA carryover between experiments and users with a system like Sony's. So I know we just sort of brushed on a lot of topics here. I confess I am not a transcriptomics workflow expert, but Sony does have a lot of those in its employ. And we have an entire subpage dedicated to genomics and how our sorters have been integrated in all sorts of experiments in this area. So there are application highlights and then a growing list of publications from leading journals, as well as some new white papers that Sony has put out. And that's all I had for you guys today. Let's check the chat to see what questions we might have.
If anyone would like to unmute themselves, you can go ahead and ask a question as well, but you can also type into the chat if you prefer to do that.
Okay, I see Brian Perchala. How good is the yield? So yeah, every sort is kind of a three-way tug of war between purity, yield, and how much time you're willing to spend pursuing either of those goals. Sony Cell Sorters can give yield as well as purity in the high 90s. So it's just a matter of sort of taking the time beforehand to optimize things from your volume that you're sampling out of your cell concentration, and then the speed at which you feed the samples. So because within each chip size, you do have the ability to, there's a little slider, like a 1 to 10 scale, and sort of the gas pedal to feed the sample through relatively slowly, or to kick it up to as fast as you can go without starting to have, you know, coincidence.
While we're waiting for some more questions, I can go ahead and share though, especially for any users there that are at Bloomington, we do have a 10x Chromium controller located in Ken Nephew's lab, which is a couple floors above the flow facility. So if anyone wanted to do 10x or single cell genomics there in Bloomington, it would just be a short walk from the flow cytometry facility up to where the controller is. And I will post a link in the chat that will, so that you can coordinate that, that kind of an appointment.
But bottom line, I would just encourage people to go see Christiane and book some hours on her SH800. In my own time at the bench, sorting was this scary thing that was just the domain of people who had been doing it for years. You almost had to be born into it. I remember in doing analysis, just using like an LSR2 and trying to compensate like 8 or 10 colors, and you know it felt like juggling that many balls. And sorting was, you know, with all the manual inputs that were required years ago, would be like, you know, juggling that many balls while trying to ride a unicycle. Sony has done a good job of sort of taking the unicycle out of the equation. And really the juggling is a lot easier too. So running one of these sorters feels very much like running an analyzer. There are boxes that you can check to have the instrument monitor your drop drive and break off. And with a 10-liter tank on this thing, you can run the entire day without having to mess with your side streams or anything. So folks that have run, like I said, an ARIA or you know, an older fixed nozzle instrument, I think we'll have a you know, good appreciation for just how rare that is to be able to run the whole day without having to think about the, you know, the instrument at all, just other than, you know, drawing your gates and you know, starting and stopping each tube. But you don't have to recalibrate the instrument. You don't have to provide manual inputs to keep getting good data out of it.
Jerry, I wonder if you could comment on the features of this sorter versus other types of sorters for sorting cells or nuclei for single cell transcriptomics. Is it the slide that you're using that makes it superior, or?
Yeah, I would say there are a couple different areas. So the first would be just simply the ease of use. You know, whether a system has a technical edge versus another, I guess doesn't really matter if you can't figure out how to use the system in the first place. So having it be so user friendly and so automated definitely does make a big difference. But yeah, having that that sorting chip that kind of looks like a microscope slide, that is a technical edge for a variety of reasons. One of the biggest is that it eliminates carryover between you know, tubes in an experiment and between experiments themselves, because you know you're starting fresh with a new chip each day, and you can actually switch between chip sizes within a day, and then go back to what you were using in the morning. So if somebody comes in early afternoon with some you know, neuronal sample or stem cells or whatever, you can switch from the 100 micron that you might have started the day with, move to the 130 micron, and then switch back. So, but yeah, having that chip be so easily disposable, and then also having a sample line that can be removed and so easily replaced is another advantage as well. It's typically not necessary. The cleaning functions of the instrument are sufficient to clean both the chip and the sample line. But as I mentioned earlier in the talk, if you're just on a really critical experiment, you know, maybe that last experiment before a thesis defense or before a submission for publication, and if it's been a couple of months since the sample line has been changed, it might be worth the $250 $30 odd that it would cost you to put in that new sample line. So, and most of the things that you would have to do with the SH800, certainly on a daily basis, and even on a monthly or semi-annual basis, those are all things that the user can do themselves. Sony has a principle called mindful design. And so that's is a real advantage when using our instruments, that we put everything up front, that we don't have fittings secured by tiny little Allen wrench headed screws or anything like that. You can tear out the sample path. You can switch around the optical filters. It's really easy to use and really easy to adapt to your particular experiment, whether that's the filter setup, or whether that's sampling out of and depositing into one particular kind of container to keep the volume down for later on when you're going through your transcriptomics workflow.
Are there any other questions? Feel free to unmute yourself if you'd like to ask.
Has anybody used the SH800?
Let's see, is Christiane still on the chat, or is still in the Zoom?
Yep, I'm still in the Zoom.
Oh, okay.
I was looking through the list to see if anybody on here has been one of the people I've trained on the instrument, but I don't think we have anybody who's been trained on it yet besides myself. So, but I will point out, so I mentioned on the website that we have information about the SH800 and other instruments, and I'm also going to put my contact information in the chat as well. You can always feel free to contact me. I'm always happy to answer any questions. But as Jerry was pointing out, it's a very easy to use or user-friendly instrument, and we have those different chip options. One of the things that in the operator, that there's a big long operator's guide, but that is mostly pointing out a lot of things are done through dialogues on the instruments. So that helps with the setup as well, and it being user friendly. And we are looking at, so again, our instrument has 2 lasers, 488 and 561 lasers, and we do have the 3 chips available to sort with, and a lot of those options that Jerry mentioned, sorting into plates, into tubes, and because we have an ARIA sorter as well, BD, and with that one, as it doesn't have that automation, and it has 14 colors, but I will be the one that will help you with that instrument. And again, I'm always happy to help, but if you have an experiment that we can help you with, and that you want to get your hands on the sorter as well, that SH800 is going to be something that's very, very useful, and again, user friendly. And I was looking at, so there was a question regarding the number of cells, and I was looking for it somewhere in the operator guide, but it does give us, and I haven't posted it to the website yet, but it gives us on that scale of 1 to 10 how many cells we can analyze and sort per second, depending on the concentration of the cells. And if I can find it, I'll share it. But the operator's guide so the big manual is 262 pages. So I don't know, Jerry, if you know what page that's on.
I don't have it on the top of my head, but you're absolutely right. It's very user friendly. I would add that, the user's manual is over 250 pages, but it's not like all single space text. There are a lot of pictures and screenshots, and with an international audience, Sony has done its best to try and make things very, very clear. So detailed processes in the owner's manual, like for instance, doing a big ethanol clean if you're going to shut down for about a month around the holidays. That might take up like 15 pages, but it's 10 of the pages are pictures and then captions off to the side or underneath. When we go and do an installation, when somebody buys an instrument, we've actually boiled down the owner's manual to 6 or 7 pages, single-sided pages, and we'll laminate them and then put them on a ring and sort of punch a hole and leave them sitting alongside the instrument. So whether you're starting up doing your calibration, whether you're trying to figure out how to set up your experiment in the software, or whether you're trying to figure out how to properly shut down at the end of the day, you don't need a 260-page manual, really. For most day-to-day operations, assuming the instrument's been well cared for and nothing untoward is happening, 6 pages can get you through your experiment. It's really nothing to be afraid of, folks.
And I didn't mean to say that it should be daunting that the user manual is that big, but as Jerry pointed out, it's a lot of pictures and diagrams and showing you what the automation looks like. Do you mind if I pop up the user manual?
Yeah, let me stop.
Yeah, I've got it up there. So let me find where that went. Too many windows open. Give me a moment.
But Sony is able to resolve about 60% of our calls for help, whether that's help me figure something out or help me fix something. We're able to do that just remotely connecting.
Yeah. And so this was the relationship between sample pressure and the sample flow that I was talking about. So that this gives you, so there was a question about number of cells, and it gives you an idea what the flow rate is and what the sample concentration is and how many events per second we're going to see with the different chips. So which is useful when you're processing a sample and bringing a sample to get an idea of what we're going to see when we put that on the instrument. But I will point out, and then let me skip to a different page, maybe one. Yeah, so there's different, as Jerry was saying. So this has a lot of diagrams that show us what's happening on the instrument, and it gives a lot of information that you don't necessarily need on a day-to-day basis, but if you're curious about, so sometimes people are wanting to dive in a little bit more, but a lot of it, as Jerry was saying, and this is probably, so these are the window descriptions. I didn't hit on the right chapter. Let's go to one, like for instance, analyzing the index sort data, what you were talking about before. It just gives that description of how that's done. And like you said, a lot of different windows to show those diagrams. So I think that's really nice as well. So yeah, so let's say, for example, if somebody was using it after hours and had a question, they could easily get to this and go, oh, okay, this is the question that I have, and it's right there for you. So,
absolutely.
Yeah.
So I did want to make one last sort of adjacent but slightly off-topic point. Besides sorters and analyzers, Sony has a growing portfolio of reagents, not just viability or proliferation dyes, but fluorochrome conjugated monoclonal antibodies, the same clones and the same colors as the BDs and the BioLegends of the world. So I put my email address and my phone number in the chat there. Sony doesn't do the volume that some of these other companies do. So I'll start with the caveat that a lot of times we're like at least $20 a tube more expensive. I don't necessarily expect people to switch to us wholesale, but in these times of supply chain disruptions and back orders, it's always good to have another source for what you need. So if I can be of help, certainly reach out to me.
Okay, well, if there's no further questions, I'd like to thank Jerry and Christiane again for presenting today, and everyone have a happy new year and happy holidays.
Happy holidays, everybody.
Great. Thank you. Happy holidays.